There's, this expectation of absolute geometric precision. You break your arm, the X-ray shows that jagged white line, and the physician just points and says, well, there it is.
Right, broken or not broken.
Exactly. The binary nature of it is comforting. We like things to be visible and easily categorized. But you step into the world of molecular diagnostics, and suddenly that X-ray machine is fundamentally broken. You're dealing with invisible molecules in this microscopic soup.
A very chaotic soup.
Yeah. And the only way to map that soup is to force dozens of highly volatile chemical reactions to happen in the exact same drop of fluid at the exact same time without them destroying each other. That is the paradox we are looking at today.
It really is the ultimate diagnostic bottleneck. I mean, you're searching for a specific sequence of nucleotides in a background of billions, and you need to find several different targets simultaneously, and often from a heavily degraded or minuscule sample, too.
Right. Which brings us to our mission for you today. We are taking a deep dive into the source material. We've got a highly comprehensive, highly technical presentation deck focused entirely on the mechanics, the chemistry, and the clinical application of multiplex PCR.
And we are going through it page by page.
Page by page, leaving absolutely nothing out. We're skipping the high school biology recap, so if you're here, you already understand the fundamentals of DNA amplification. We're jumping straight into the deep end of the thermodynamics, the stoichiometric balancing acts, and the bioinformatics that make multiplex PCR possible.
Yeah, because the source deck is incredibly rigorous. It traces the operational logic from the initial 1988 breakthrough all the way to the complex multiplex arrays that drive modern epidemiological surveillance. It treats the reaction tube not just as a biological environment, but as a strict biophysical engine.
Okay, let's unpack this. We'll start right where the deck begins, with a 10,000-foot view. It notes that multiplex PCR was first reported back in 1988. It defines the technique strictly as the simultaneous amplification of two or more target sequences by including more than one pair of primers in the same reaction mixture.
Exactly.
So we are moving from a paradigm of many separate, isolated reactions to a single run in a single vessel from a single sample aliquot.
And that 1988 origin point is critical context. I mean, this was an era when the polymerase chain reaction was still relatively new. The sheer labor of running individual assays was paralyzing laboratories.
I can imagine.
Right. So the conceptual leap wasn't just about saving time, though that was huge. It was about data integrity. If you run 5 different targets and 5 different tubes, you introduce 5 different variables of pipetting error, thermal fluctuation, contamination risk.
Oh, that makes sense.
Yeah. By forcing them into one single sample aliquot, you lock the variables. If the reaction works, it works for everything under the exact same parameters.
So the deck provides this visual to anchor this, titled Traditional PCR vs. Multiplex PCR. The traditional model shows a linear, isolated process. Sample 1 goes into a tube with primer set 1, sample 2 with primer set 2, and so on.
You end up with 4 distinct plastic tubes.
Right. But the multiplex model shows all 4 of those samples and all 4 of those distinct primer sets just funneled down into one single test tube. So traditional PCR is like cooking every side dish for Thanksgiving in a different oven, while multiplex PCR is like tossing all the ingredients into one magical slow cooker that perfectly cooks everything at once.
That's a great way to picture it.
But doesn't that make the cooking process chaotic? I mean, throwing everything in one pot.
Oh, absolutely. That captures the inherent volatility perfectly. You are dealing with microscopic thermodynamics and stochastic collisions. When you put multiple primer sets into one environment, they do not politely wait their turn.
They don't form a line.
No line. They are constantly colliding, annealing, detaching based entirely on localized kinetic energy. If primer A has a higher binding affinity than primer B, it'll just dominate the free tack polymerase in the solution.
Which means we have to impose order on that chaos.
Yeah.
The document outlines the rules of the game. It lists 4 non-negotiable criteria for a successful multiplex reaction.
Right, the guardrails.
Yeah. First, the primer design for all pairs has to be optimized so that they can all work at the same annealing temperature.
And this is where the biocomputational design becomes brutal. The annealing temperature, or TEMM, is dictated by the precise ratio of guanine-cytosine to adenine-thymine bonds in your primer sequence, along with the overall length and the salt concentration of your buffer.
So it's a math problem.
Exactly. You cannot just arbitrarily tell 20 different primers to bind at 60 degrees Celsius. You have to reverse engineer the sequences of your target pathogens or genes to find specific regions that naturally possess matching thermodynamic profiles.
So what does this all mean for the physical tube? Like if you have one primer set that naturally binds at 65 degrees and another that binds at 55 degrees, what physically happens when the thermal cycler drops to 60 degrees?
Well, the 65-degree primer finds its target and locks on immediately because the kinetic energy is already low enough for its hydrogen bonds to stabilize. So the Taq polymerase recognizes that double-stranded complex and begins extending.
While the other one is just floating there.
Exactly. By the time the machine drops down to 55 degrees to allow that second primer to finally stabilize and bind, the first target has already monopolized the available DNTPs.
The loose nucleotides.
Right, the loose bricks. And it's occupied a massive percentage of the enzyme. So the first target amplifies exponentially, while the second target is starved of resources and barely amplifies at all. You end up with severe amplification bias.
Wow, okay. And the second rule states the primer pairs must be highly specific. We aren't just looking for close enough. If a multiplex tube contains dozens of oligonucleotides, a lack of specificity doesn't just mean you amplify a harmless background sequence, right? It means the primers start cross-reacting.
And cross-reactivity in a multiplex assay is fatal to the data.
Fatal.
Yeah. If a forward primer for, say, influenza A accidentally shares 8 complementary bases at its 3' end with the reverse primer for RSV, they will hybridize to each other. Taq polymerase will then blindly extend them, creating a completely artificial, incredibly short piece of DNA called a primer-dimer.
A primer-dimer.
Right. And because it's so short, it amplifies with terrifying efficiency, completely outcompeting the actual viral targets.
That feeds directly into the third rule. Amplicon size shouldn't differ too much. The amplicon being the final, copied piece of double-stranded DNA. Why does the final physical size of the product dictate the efficiency of the reaction?
It comes down to the processivity of the Taq polymerase. Processivity refers to how many nucleotides the enzyme can add before it inevitably just falls off the template strand.
Okay.
If you're multiplexing a 150 base pair target alongside a 2000 base pair target, the enzyme completes the 150 base pair sequence in seconds and immediately moves on to the next one. The 2000 base pair target requires the enzyme to stay attached and working for a significantly longer duration.
So the shorter one just wins the race.
Every time. In a competitive environment with limited resources, the shorter amplicon will always outcompete the longer one due to sheer kinetic turnover. Keeping the amplicon sizes relatively similar ensures a level playing field.
And the fourth rule ties that all together, which is setting the polymerization time optimally. You have to give the chemical machinery exactly enough time to copy the longest fragment in the mix without wasting time overcopying the shorter ones. So if we look at the constraints of these 4 rules, matching thermodynamics, extreme specificity, unified amplicon sizing, and optimal extension times, it forces us to evaluate the trade-offs. The source document explicitly weighs the advantages against the disadvantages.
It's quite honest about it.
Yeah, it lists the primary advantage as producing considerable savings in time and effort within the laboratory without compromising the utility of the experiment.
Right, because in a high-throughput clinical laboratory or, a forensic facility, hands-on technician time and thermal cycler real estate are the ultimate bottlenecks.
Time is money.
Exactly. If you're screening a cohort of 500 patient samples for 5 different mutations, running singleplex reactions requires 2,500 individual PCR setups.
That's absurd.
It's a nightmare. Multiplexing condenses that back down to 500. The throughput scaling is nonlinear. It completely transforms the operational capacity of the facility.
But the disadvantages slide reads almost like a warning label. First bullet point. Optimization is difficult, since many sets of forward and reverse primers are to be designed for use. Second point, increases cost. Wait, it says it saves time and effort, but then immediately says optimization is difficult and increases cost. How do we reconcile that?
Well, it's the classic distinction between upfront setup costs versus long-term operational savings. Designing a multiplex PCR assay from scratch is an agonizingly expensive and failure-prone process.
Because of all those rules we just talked about.
Exactly. You are paying for months of bioinformatic analysis, purchasing dozens of highly purified, custom-synthesized oligonucleotides, and burning through expensive reagents running hundreds of gradient optimizations to force these disparate molecules to cooperate.
So the R&D phase is a massive sink.
Huge. However, once that assay is validated and locked in, the cost per test drops exponentially. You pay a heavy toll to build the infrastructure, but the subsequent transportation is incredibly cheap.
The document also re-emphasizes the chemical danger here under the disadvantages. It says, presence of multiple primers may lead to cross-hybridization with each other and the possibility of mispriming with other templates. We touched on primer dimers, but mispriming with other templates suggests the primers are actually attacking the wrong genomic DNA.
Yeah, they are. When you extract DNA from a clinical sample, say a buccal swab, you aren't just getting the viral DNA you are looking for, you are extracting the entire human genome, bacterial flora, and degraded cellular debris.
It's a mess in there.
It's a massive, complex background of non-target nucleic acids. With 20 different primers floating in the mix, the statistical probability that one of them will find a pseudo-complementary sequence in that vast human genomic background increases dramatically.
And if it binds to the human genome.
It initiates amplification, wastes resources, and creates artifact bands on your final gel. It just muddies the waters.
Having established those severe stakes, the source material pivots to the architectures. The actual physical configurations of multiplex PCR. It splits them into two distinct categories, single template PCR and multiple template PCR.
Right.
Let's break down the single template architecture first. The text notes this technique uses a single template, which can be genomic DNA along with several pairs of forward and reverse primers to amplify specific regions within that single template.
This is primarily utilized in human genetics and oncology. You possess a single source of DNA, which is the patient's genome. You aren't looking for foreign invaders. You're interrogating the patient's own code for multiple distinct structural anomalies.
Okay, can you give an example?
Sure. Duchenne muscular dystrophy is often caused by large exon deletions within the dystrophin gene. Instead of sequencing the entire massive gene, a single template multiplex assay uses primers flanking the most commonly deleted exons. If the exon is present, you get a band. If the exon is deleted, that specific band is absent from the final readout.
Got it. Now, the multiple template PCR operates on a different logic. It uses multiple templates and several primer sets for each template in the same reaction tube. The visual graphic on this slide is incredibly detailed.
It really is.
It shows 4 distinct sources at the top, strain A, strain B, strain C, and strain D. Each strain has its own circular coiled DNA. Arrows point down, indicating that the DNA from strain A receives a specific red primer set, strain B, a blue set, strain C, a green set, and strain D, a purple set. All of these.
Everything.
Everything. The 4 bacterial strains, the 4 primer sets, all funneled into one single plastic PCR tube. Then there's an arrow pointing down from that tube to a barcode-like readout labeled marker. It shows distinct separated bands at specific base pair lengths. The green band is at 371, the red at 275, the purple at 202, and the blue at 141.
That barcode is a representation of an agarose gel electrophoresis result, and it perfectly illustrates how we circumvent the invisible nature of the reaction.
Because we can't actually see the DNA being copied.
Right, so we engineer the primers to produce amplicons of strictly defined, mathematically distinct sizes. When you run the final product through a charged gel matrix, the smaller DNA fragments, like that 141 base pair blue amplicon, migrate rapidly toward the positive electrode. The larger fragments, like the 371 base pair green amplicon, experience more physical drag in the gel matrix and migrate much slower.
So it's like having one search party looking for 4 different people in a forest versus 4 search parties looking for one person. The physical distance the DNA travels directly correlates to the specific pathogen it represents.
Exactly.
You look at the gel and see a band at 371 and a band at 141, and you can definitively state that strain C and strain D were present in the sample, while strains A and B were absent.
It's beautifully efficient.
It is, but how do we physically tune the reaction to make this happen? I mean, guarantee those distinct bands emerge without the background noise. That transitions us directly into the most rigorous section of the deep dive, the reaction mix.
Oh yeah, the chemistry.
The document provides an exhaustive, ingredient-by-ingredient breakdown of how to manipulate the benchtop chemistry to force optimization.
What's fascinating here is where the theoretical elegance of multiplexing collides with the unforgiving realities of stoichiometry. Every microliter added to that tube changes the overall kinetic environment.
Right. So the text starts with the amount of primer. It dictates that initially you must start with equimolar primer concentrations, meaning precisely the same amount of every primer, ranging from 0.1 to 0.5 micromolar. However, it explicitly states that when there is uneven amplification, you must abandon the equimolar approach.
You have to adapt.
Yeah, you must increase the amount of primers for the weak loci and decrease the amount for the strong loci. The final optimized concentration can vary wildly, from 0.04 to 0.5 micromolar. Why is one locus inherently weak while another is strong, assuming they were both designed within the same TM window?
Well, the TM window is only a mathematical average based on the primary sequence. It doesn't account for secondary structures in the template DNA. The target region for a weak locus might naturally fold in on itself, forming a tight hairpin loop that physically blocks the primer from accessing the binding site.
Even at the correct temperature.
Even at the correct annealing temperature, yeah. Alternatively, a strong locus might reside in an open, easily accessible region of the genome.
So adjusting the weak and strong loci is like adjusting the volume on a soundboard so the bass doesn't drown out the vocals. Altering the concentration is essentially a kinetic handicap.
That's a great analogy. The rate of any chemical reaction is directly proportional to the concentration of the reactants. If primer A has a massive structural advantage and binds too quickly, you starve the reaction of primer A.
Drop the volume.
Exactly. You drop its concentration to 0.04 micromolar, drastically reducing the statistical likelihood of it finding its target. Conversely, you flood the tube with 0.5 micromolar of primer B, the weak locus, relying on sheer numerical superiority to force it to bind to its stubborn, folded target. You're artificially manipulating the collision rates.
Wow. Okay, moving to the next variables. DNTP and MgCl2 concentrations. The document states that MgCl2 magnesium chloride concentration is kept strictly constant at 2 millimolar. Meanwhile, the DNTP concentration, the loose nucleotide bricks, must be increased stepwise from 0.5 to 1.6 millimolar to accommodate the multiple targets.
This is such a crucial balance.
It emphasizes that the optimization of magnesium is critical because Taq DNA polymerase is a magnesium-dependent enzyme. Let's dig into the bioinorganic chemistry here. Why does the enzyme care about magnesium, and why is the balance between magnesium and the DNTPs so volatile?
Well, Taq polymerase is a metalloenzyme. Its active site contains highly conserved aspartate residues that coordinate two magnesium ions. Those Mg2+ ions are not just structural, they are the catalytic engine. They position the incoming DNTP precisely and pull electron density away from the phosphate groups, allowing the chemical bond to form. Without magnesium, Taq is basically an inert piece of protein.
So it needs the magnesium to do the work.
Exactly. But here is the kinetic trap. DNTPs are highly negatively charged, and they act as powerful chelating agents. They strongly bind to free magnesium ions in the solution.
So if you blindly increase the DNTPs to 1.6 millimolar to provide enough raw material for 10 different multiplex targets, those extra DNTPs will chemically soak up all the free magnesium in the buffer.
Yes. You strip the magnesium out of the solution, the Taq polymerase loses its catalytic core, and the entire reaction grinds to a halt. Conversely, if you add too much magnesium, say 4 millimolar, to compensate, the Taq polymerase becomes hyperactive and loses its proofreading stringency.
It gets sloppy.
Very sloppy. It will start extending primers that are only partially matched to the wrong targets, flooding your tube with nonspecific artifacts. The 2 millimolar magnesium to 0.5, 1.6 millimolar DNTP ratio is an incredibly delicate stoichiometric tightrope.
The document then addresses the PCR buffer, noting that raising the buffer concentration to 2x improves the efficiency of the multiplex reaction. The buffer provides the ionic strength and pH stability. I imagine that with the massive amount of phosphate being cleaved from the DNTPs during a heavy multiplex reaction, the localized pH could shift drastically without a robust buffer.
It absolutely would. The 2x buffer acts as a thermodynamic shock absorber, maintaining the specific ionic environment Taq requires despite all the massive simultaneous molecular construction projects happening around it.
Then we reach the amount of template DNA and Taq. The text advises keeping the template DNA low for optimum, efficient, and specific amplification. It also suggests testing different Taq concentrations experimentally. But why keep the original template DNA low? If you're searching for a needle in a haystack, shouldn't you want as many needles in the tube as possible?
You want needles, but you are also adding the entire haystack. Clinical samples contain massive amounts of background genomic DNA. If you input too much total template, the sheer volume of non-target DNA creates a statistical certainty that your primers will find pseudo-complementary sequences and misprime.
More noise.
Right. And high template concentrations also increase the viscosity of the solution, which slows down the diffusion rate of the primers and the Taq polymerase. By drastically diluting the template, you reduce the background noise and allow the reaction to remain kinetically fluid. You force the primers to only interact with their absolute perfect thermodynamic matches.
Which brings us to the most chemically aggressive part of the mix, the adjuvants. the slideless DMSO, glycerol, and BSA. It states these additives can significantly improve the most difficult reactions because they relax DNA, thus making temperate denaturation easier.
This is some of the coolest chemistry.
I'm stuck on these adjuvants. Are they basically a chemical massage for the DNA to get it to unwind? How does a solvent like DMSO relax DNA?
Chemical massage is actually pretty accurate. Dimethyl sulfoxide, or DMSO, is a powerful polariprotic solvent. In regions of the DNA that are extremely rich in guanine and cytosine, the two strands are held together by 3 hydrogen bonds per base pair, rather than the 2 bonds found in adenine-thymine pairs.
These are locked tighter.
Much tighter. These GC-rich regions form incredibly stable, rigid structures that often refuse to separate even at 95 degrees Celsius. DMSO molecules physically intercalate and disrupt the water lattice structure around the DNA. They compete for the hydrogen bonding sites on the bases, effectively weakening the interaction between the 2 DNA strands.
Wow.
Yeah, of adding 5% to 10% DMSO, you physically lower the melting temperature of those stubborn GC-rich regions, forcing them to peel apart and allowing the Taq polymerase to access the sequence.
But doesn't that inherently destabilize the primer target annealing as well?
It does, and that is exactly why optimization is so difficult. DMSO lowers the melting temperature of the target, but it also lowers the melting temperature of your primers. If you add too much DMSO, your primers will never lock onto the target, and the reaction fails. It is a razor-thin margin between relaxing the template and destroying the primer affinity.
And what about BSA? Bovine serum albumin. It's a protein. Why add a cow protein to a multiplex DNA reaction?
BSA doesn't interact with the DNA, it interacts with the environment. In clinical samples like blood, feces, soil, there are numerous naturally occurring PCR inhibitors, like high bile salts or humic acids. These inhibitors bind to Taq polymerase and deactivate it.
So the BSA is like a shield.
More like a molecular decoy. It's a large, sticky protein that binds up those inhibitors, sequestering them away from the Taq. Furthermore, BSA prevents the Taq polymerase from sticking to the plastic walls of the reaction tube, keeping it suspended in the active solution.
We've mathematically handicapped the primers, balanced the magnesium chelation, stabilized the pH, lowered the background noise, and chemically massaged the GC-rich regions while sequestering inhibitors. The benchtop chemistry is locked. Now, we introduce the kinetic energy.
Time for the heat.
Yeah. The document details the thermal choreography. It highlights NEB's TUM calculator, providing a visual of the web interface used to estimate annealing temperatures based on complex algorithms. Then it lays out the strict process followed for 5x concentration. Let's walk through this thermal life cycle. First, initial denaturation. 95 degrees Celsius for 1 to 2 minutes. Second, a shorter cycling denaturation. 95 degrees for 5 to 30 seconds.
At 95 degrees, the thermal energy violently exceeds the hydrogen bond energy holding the double helix together. The DNA physically denatures into single strands. The initial 1 to 2 minutes is required to ensure that the massive, highly complex genomic template is completely unwound.
In the shorter bursts.
The subsequent 5 to 30 second bursts are shorter because you are only denaturing the much smaller amplicons generated in the previous cycles.
Then we hit the annealing phase, lasting 30 seconds to 1 minute. The text explicitly notes this is optimized by doing a temperature gradient PCR, starting 5 degrees Celsius below the calculated T.
Okay, I have to ask. Why intentionally start colder? Why do we abandon the mathematical certainty of the calculator? What are we trying to provoke in the reaction?
Because the algorithms calculating TM are theoretical, they assume a perfect aqueous environment. But as we just discussed, our tube is full of DMSO, 2x buffer, competing DNTPs, and massive amounts of background DNA. The physical reality of the tube alters the true annealing temperature.
Right.
By running a temperature gradient, meaning you program the thermal cycler to run different rows of tubes at 55, 56, 57, 58, and 59 degrees simultaneously, you're testing the math against reality.
Yes.
You intentionally start 5 degrees below the calculated T because lower temperatures thermodynamically favor binding.
Oh, so it forces them to stick? It forces the primers to stick to the template, even if imperfectly. You are guaranteed to see amplification, even if it's messy and nonspecific. From there, you analyze the gel and slowly raise the temperature in the next run, increasing the kinetic stringency until the background noise disappears, leaving only the perfectly matched multiplex targets. You start permissive to ensure the chemistry works, then you tighten the vise.
The next phase is extension, recommended at 68 degrees Celsius for 1 to 2 minutes per kilobase, followed by a final extension at 68 degrees for 5 minutes. Now, I want to push back on this. Every basic PCR protocol uses 72 degrees Celsius for the extension phase because that is the absolute optimum velocity for Taq polymerase. Why does this multiplex document specifically demand 68 degrees?
That is a phenomenal detail to catch. While 72 degrees is optimal for sheer speed, it is kinetically aggressive. In a highly complex multiplex reaction, especially one amplifying large fragments or utilizing multiple primer sets, 72 degrees can be too hot for the primers to remain stably bound to the template while the Taq initiates extension.
It just melts them right off.
Exactly. The thermal energy begins to melt the primer off the template before the enzyme can secure it. By dropping the extension temperature to 68 degrees, you sacrifice a slight amount of enzyme velocity, but you massively increase the structural stability of the primer template complex, drastically reducing the drop-off rate and increasing the overall yield.
The choreography repeats for 30 to 35 cycles.
Mm-hmm.
We've mastered the environment and the thermodynamics, but the actual targeting system, the primers themselves, requires immense precision. The document moves into the primer design parameters, stating that specific design is the absolute foundation for high yield. Let's dissect the 4 parameters. 1. Primer length. It dictates an appropriate length, usually short, in the range of 18 to 22 bases. Why is this the mathematical sweet spot?
It is an exercise in sequence entropy. The human genome is over 3 billion base pairs long. A sequence of 18 to 22 nucleotides provides enough unique combinatorial diversity, that's 4 to the power of 20, that the specific sequence should theoretically only exist exactly once in the entire genome.
And if it's shorter?
If you go shorter, say 12 bases, the sequence might randomly appear thousands of times across the genome, destroying your specificity. If you go longer, say 35 bases, the primer develops complex secondary structures. It begins folding back on itself, forming internal hairpins that prevent it from binding to the target.
Parameter 2 is the melting temperature, or TIM. It advises aiming for a similar TIM across the pool, preferably between 55 and 60 degrees. For sequences with high GC content, a higher TIM of 75 to 80 degrees is recommended. Crucially, it accepts a TIM variation of between 3 to 5 degrees for primers used in a single pool.
That 3 to 5 degree window is the pragmatic concession to the reality of bioinformatics. It is mathematically impossible to find 20 unique, highly specific 20-mer sequences across 5 different viral genomes that all possess the exact same theoretical melting point.
Nature doesn't work like that.
Right. The 3 to 5 degree variance is the operational tolerance that the temperature gradient optimization is designed to smooth over.
Parameter 3 demands specificity, citing the fierce competition in a single vessel. But parameter 4 is where the chemistry turns hostile. Avoid primer-dimer formation. The text states, the design primers should be checked for formation of primer-dimers, with all the primers present in the reaction mixture. Dimerization leads to unspecific amplification. We touched on this, but let's look at the thermodynamics of a primer-dimer.
Yeah.
Why are they so destructive?
A primer dimer is the ultimate parasitic amplicon. Because primers are added at micromolar concentrations, they vastly outnumber the actual target DNA in the initial cycles. If the 3' end of the forward primer happens to have 3 or 4 bases that complement the 3' end of a reverse primer, they will transiently hybridize when the temperature drops during the annealing phase.
And Taq just sees a job to do.
Taq polymerase does not know what it is copying. It only recognizes a double-stranded sequence with an available 3' hydroxyl group. It grabs that transiently hybridized primer complex and extends it. You now have a permanent, perfectly complementary piece of DNA that is only 40 base pairs long.
Oh, wow.
And in the next thermal cycle, this tiny 40 base pair parasite melts and acts as a massive template sink. Because it is so short, the enzyme copies it in milliseconds. It consumes your DNT piece, chelates your magnesium, and exhausts your Taq polymerase within 10 cycles, leaving your actual viral targets completely unamplified.
The text notes that these parameters are heavily aided by computational software, but eventually you have to run the physical test. And that brings us to detection. The document notes that for fluorescent dye-based detection, CyBr green is the most commonly used agent. Here's where it gets really interesting. It explains the mechanism. The CyBr green dye functions as an intercalating agent that emits detectable fluorescence when bound to double-stranded DNA. Imagine that like a glow stick fluid that only lights up when it gets wedged between the rungs of the DNA ladder. Does this mean we can actually see the success in real time?
We can. CyBr green is an asymmetrical cyanine dye. When it is floating free in the aqueous buffer surrounded by water molecules, its fluorescent yield is incredibly low. It is essentially dark. However, the molecule is flat and planar.
Like a disc.
Yeah, exactly. During the extension phase, as Taq polymerase builds the new double-stranded DNA, the cyber green molecules physically slip or intercalate into the minor groove of the DNA double helix, wedging themselves between the stacked nucleotide base pairs.
That's incredible.
Once trapped inside the rigid structure of the DNA, the dye is shielded from the water. Its molecular rotation is locked. When the thermal cycler's laser hits it with blue light at 497 nanometers, it absorbs the photon, cannot dissipate the energy through physical vibration, and is forced to emit a highly intense green photon at 520 nanometers.
So in cycle 1, you have almost no double-stranded DNA, so there's no intercalation, and the tube is dark. By cycle 20, you have a billion copies of double-stranded DNA, billions of cyber green molecules lock into the minor grooves, and the tube intensely fluoresces green. A camera records that optical emission after every single cycle, giving you a real-time kinetic curve, the amplification. You are watching the biomachinery operate live.
It's beautiful.
With all of the thermodynamic and optical parameters established, the document shifts entirely. It moves into the hidden superpowers of the technique, advantages that go far beyond saving physical laboratory time. The first advantage listed is the utilization of internal controls. The text states, potential problems in a simple PCR include false negatives due to reaction failure. False negatives are often revealed in multiplex assays because each amplicon provides an internal control for the other amplified fragments. Let's examine the clinical statistics of a false negative. If I run a test and it's blank, I don't know if I messed up the test or if the target isn't there. Multiplexing is like having a canary in the coal mine alongside the actual test.
That's exactly it. An ambiguous negative is the most dangerous result in diagnostics. Is the tube blank because the patient is genuinely free of the virus? Or is it blank because the DNA extraction failed, the technician forgot to pipette the Taq polymerase, or the thermal cycler block malfunctioned? If you report that ambiguous negative to the physician, you might be sending a highly infectious, critically ill patient back into the population.
But multiplexing inherently solves this. You don't just test for the virus.
Right. In a multiplex clinical assay, you engineer one of your primer sets to target a highly conserved housekeeping gene that is universally present in every human cell, such as the RNase P gene or beta-actin. That is your internal positive control.
So if the test worked, that gene should always show up.
Always. When you run the multiplex, you look at the real-time optical curve. If you see the strong fluorescent signal for the human RNase P gene but zero signal for HIV, the ambiguity is destroyed. You know with absolute thermodynamic certainty that the DNA extraction worked, the Taq was active, the thermal cycling was perfect, and the primers functioned. The assay was robust. Therefore, the absence of the HIV signal is a true definitive negative.
And if the tube is completely dark,
No HIV, no RNase P, you immediately know the assay chemistry failed and the sample must be reprocessed.
the document also highlights efficiency, conserving costly polymerase and rare templates. In neonatal diagnostics or neuro-oncology, you might only receive a microscopic volume of cerebrospinal fluid or a fragmented biopsy. You cannot physically split that sample into 10 different tubes without diluting the DNA below the limit of detection. Multiplexing extracts 10 dimensions of data from a single, undiluted drop.
That conservation is critical in the clinic.
It further lists indication of template quality and template quantitation. It notes that by mathematically accounting for reference templates, amplification cycles, and inhibition rates, you can assess the exact quantity of the target.
Because we are using intercalating dyes or specific fluorescent probes, we aren't just looking at an endpoint gel. We are looking at the exact cycle number where the fluorescence crosses the background threshold, the seat value.
Seed value.
Yeah. By comparing the seat value of your viral target against the seat value of a known titrated standard curve running in the same multiplex reaction, you can calculate the exact viral load. You shift the clinical diagnostic from a qualitative, is the virus present, to a highly actionable, how many viral copies per milliliter are in the patient's blood.
Which bridges us directly into the ultimate application of this technology, the real-world impact. The source document provides an incredibly dense, exhaustive breakdown of the applications of multiplex PCR, moving from theoretical chemistry to clinical deployment. It lists 10 broad categories: pathogen identification, high-throughput SNP genotyping, mutation analysis, gene deletion analysis, template quantitation, linkage analysis, RNA detection, and forensic studies.
It covers almost every major field of molecular biology.
It anchors this with two critical timeline notes. In 2008, multiplex PCR was used for analysis of microsatellites in SNPs, and significantly, in 2020, RT-PCR multiplex assays were designed that combined multiple gene targets from the Center for Diseases and Control. Let's examine that 2020 application. The global response relied entirely on multiplexing, but the text specifies they combine multiple gene targets of the same virus. Why multiplex different parts of the same genome?
It was a masterclass in evolutionary surveillance. RNA viruses mutate at a staggering velocity. If you design a singleplex assay targeting the spike protein gene and the virus undergoes a dilution mutation in that exact sequence, your primers will no longer bind.
And the assay goes blind.
The virus is still there, still replicating, but it produces a false negative. By utilizing an RT-PCR multiplex assay that simultaneously targets the spike gene, the nucleocapsid gene, and the envelope gene in a single tube, you create a robust diagnostic safety net.
Oh, I see.
Yeah, if the spike gene mutates and drops out of the assay, the optical curves for the nucleocapsid and envelope genes still fluoresce. The assay still positively identifies the pathogen while simultaneously alerting epidemiologists that a structural mutation has occurred.
Let's plunge into the massive tables provided in the document. Table 1 outlines applications of multiplex PCR to the diagnosis of infectious diseases, categorized strictly by agent. Under viruses, it lists multiplexing for HIV-1 and 2, HTLV-1 and 2, and a panel for HSV, VZV, CMV, and EBV targeting the central nervous system.
A huge array of pathogens.
Under bacteria, it lists targets like Haemophilus influenza, Streptococcus pneumonia, and mucosal pathogens like Campylobacter, Neisseria gonorrhea, and Chlamydia trachomatis. Under parasites, it even targets waterborne pathogens simultaneously, Giardia lamblia and Cryptosporidium parvum.
It's extremely comprehensive.
But Table 2 is where the true clinical logic reveals itself. Table 2, application for diagnosis of viral infections, categorizes not by biology, but by clinical manifestations. Meningitis, respiratory infections, genital lesions, vesicular rashes. Looking at these tables, it's basically a menu of human ailments. What stands out to you about the fact that one single technique can map a genital ulcer, a respiratory infection, and a parasite from drinking bad water? Why is grouping by clinical manifestation so revolutionary compared to the singleplex approach?
Because it aligns the molecular chemistry with the reality of emergency medicine. Consider the vesicular rash panel. A patient presents with severe blistering lesions. To a physician's naked eye, it could be herpes simplex virus 1, herpes simplex virus 2, varicella zoster virus, or even a localized enterovirus. The clinical presentations overlap heavily, but the antiviral treatments and isolation protocols are completely different.
So you have to guess.
In a traditional singleplex workflow, the physician has to play a guessing game based on probability. They order a test for HSV-1. 2 days later, it comes back negative. They order a test for VZV. 2 days later, negative. By day 6, the patient has suffered irreversible tissue damage.
That's terrifying.
With syndromic multiplex panels, the physician doesn't have to guess. They swab the lesion, send it to the lab, and request the vesicular lesion panel. The lab extracts the DNA and runs a single multiplex tube containing perfectly optimized primer concentrations, carefully balanced magnesium, and precise thermal gradients that searches for HSV-1, HSV-2, VZV, and Treponema pallidum simultaneously. Within 3 hours, the optical readout pinpoints the exact pathogen.
The presentation visualizes this clinical workflow perfectly. It provides a flowchart starting with a graphic of a nasal swab, which moves to a tube for RNA isolation and then down to cDNA generation.
Let's pause on that step, because the document explicitly notes RT-PCR here. Standard Taq polymerase cannot read RNA. Correct. The vast majority of acute respiratory pathogens, influenza A, influenza B, RSV, rhinoviruses, are RNA viruses. Their genomes are single-stranded RNA. The Taq polymerase enzyme is strictly a DNA-dependent DNA polymerase. It is blind to RNA.
So you have to translate it first.
Exactly. Before the multiplex PCR can even begin, the extracted viral RNA must be exposed to an entirely different enzyme, reverse transcriptase. This enzyme binds to the RNA and synthesizes a perfectly complementary strand of DNA known as cDNA. This preliminary reverse transcription step must occur in the exact same tube, requiring its own thermodynamic optimization before the thermal cycler ramps up to 95 degrees to denature the cDNA and begin the multiplex amplification.
So the RNA is translated into cDNA. The flowchart then shows a massive red arrow pointing down to a glowing PCR thermal cycler machine. A bracket opens up, showing the internal multiplex reaction. Primer set 1, hunting for virus A, primer set 2 for virus B, primer set 3 for virus C, and primer set 4 for virus D. The final visual shows the gel electrophoresis readout for 3 different patient lanes. Patient 1 displays bands for virus C and D. Patient 2 displays bands for virus B and D. Patient 3 displays only a band for virus C.
So in a single thermal cycling run, utilizing one master mix, the laboratory conclusively diagnosed 3 different patients presenting with overlapping, highly ambiguous respiratory distress.
It is the pinnacle of diagnostic efficiency.
It is, but as we look at that elegant, clean gel readout, we have to mentally superimpose the grueling chemistry we spent the last hour discussing. The only reason those bands are sharp, the only reason there are no primer-dimer smears at the bottom of the gel, and the only reason the 1.6 millimolar DNTPs didn't strip the magnesium from the talc is because a bioengineer spent months agonizing over the stoichiometric ratios.
Which introduces the ultimate irony of the entire presentation deck. After spending 15 pages exhaustively detailing the brutal manual balancing act of magnesium chelation, DMSO adjuvants, sequence entropy, and thermal gradients, the document ends with Section 10, off-the-shelf solutions. It reveals the existence of commercial multiplex kits.
If we connect this to the bigger picture, it's the commercialization of the multiplex paradigm. It is the inevitable trajectory of any complex biotechnology.
The slide lists several specific examples. First is the QIJ Multiplex PCR kit. It explicitly boasts that it comes with a 2x multiplex PCR master mix, providing a final concentration of exactly 3 millimolar MgCl2. It includes proprietary Q solution and a built-in hot start. The bullet point proudly advertises, no optimization required.
Famous last words.
Then it lists the Platinum Multiplex PCR Master Mix, designed specifically for endpoint PCR. It claims the capacity to amplify up to 20 amplicons in a single reaction, handling varying fragment sizes from 50 base pairs up to 2.5 kilobases, while maintaining high specificity and an absence of primer dimers. Finally, the 2x Multiplex Hot Start PCR Master Mix from Biotech24, promising exceptionally pure tack and fast setup. So after teaching us how to build a car from scratch, the document says, by the way, you can just buy a sports car at the dealership. Why did we need to learn all the optimization parameters if these kits exist?
Because the claim of no optimization required is a marketing statistical average, not a biological absolute. These commercial kits are undeniably brilliant. The chemical engineers at Kirigen and Biotech24 have developed highly proprietary buffer formulations. Their Q solution is likely a highly refined, patented derivative of the DMSO and betaine adjuvants we discussed earlier, designed to universally lower the melting temperatures of GC-rich templates without destabilizing primer affinity.
So they do the heavy lifting for you.
They provide a highly buffered, extremely forgiving thermodynamic environment. For 80% of standard laboratory applications, you can simply add your template, add your 20 primers, and the kit will brute force the reaction to success. But biology is infinitely complex. When you are operating at the absolute edge of detection, perhaps attempting to multiplex a highly degraded forensic bone sample at a highly mutated oncology panel, the automatic transmission will fail.
And what happens then?
The proprietary buffer will encounter a secondary structure it cannot relax. The standard 3 millimolar magnesium will be insufficient for the specific chelation dynamics of your highly concentrated primer pool. When the commercial kit fails and you get a massive primer-dimer smear or complete signal dropout, you are entirely blind if you do not understand the underlying biophysics. You cannot troubleshoot a black box.
So you have to know how it works.
The knowledge of how magnesium interacts with the TAC active site, or how a 5-degree temperature gradient alters hybridization kinetics, allows you to open up that commercial kit, alter the stoichiometry manually, and force the reaction to function. Foundational knowledge is the only diagnostic tool when the shortcut breaks down.
That structural understanding is vital. I want to examine one specific chemical modification highlighted in all of these kits. The Kirigen and the Biotech24 kits both heavily advertise a built-in hot start. What exactly is the kinetic mechanism of a hot start, and what problem is it solving in the workflow?
It solves the problem of room temperature thermodynamics. When a laboratory technician is pipetting the primers, the template, the DNTPs, and the TAC polymerase together into the tube on their benchtop, the ambient temperature is roughly 25 degrees Celsius. At 25 degrees, primers will indiscriminately bind to almost anything.
You just grab onto whatever is close.
No, exactly. The sequence stringency is completely lost. They will bind to each other, forming primer dimers, or they will loosely bind to a random background human DNA. The catastrophic issue is that wild-type TAC polymerase retains a small amount of catalytic activity even at room temperature.
So it starts building before you even put it in the machine.
Yes. While the technician is walking the tubes across the lab to the thermal cycler, the Taq polymerase is slowly, blindly extending those completely mismatched primers. By the time the machine turns on, the tube is already polluted with millions of non-specific artifacts.
So how do you chemically enforce a hot start?
You paralyze the enzyme. The manufacturers utilize monoclonal antibodies or specific chemical moieties that physically bind to the active site of the Taq polymerase. At room temperature, the enzyme is completely locked. It cannot interact with the DNTPs or the template.
That's brilliant.
The technician can leave the fully mixed tube on the benchtop for hours, and the chemistry remains completely inert. The reaction only initiates when the tube is placed in the thermal cycler and hits the initial denaturation phase at 95 degrees Celsius.
Which burns off the lock.
Exactly. That intense thermal energy permanently denatures the paralyzing antibody, exposing the active site of the Taq polymerase. By the time the enzyme is active, the temperature is so high that all the nonspecific low-affinity primer interactions have melted away. The enzyme is released into a pristine kinetic environment, guaranteeing absolute specificity from cycle 1.
It is a stunning piece of molecular engineering.
Yeah.
We have dissected every page of this document. We've traced the evolution from the isolated single-plex reactions of 1988 into the chaotic, highly competitive environment of the multiplex tube. We explored the rigid biophysical constraints required to govern that chaos.
The precise matching of sequence entropy for annealing temperatures?
Right, and the careful limitation of amplicon sizes to respect enzyme processivity, and the delicate stoichiometric balancing of magnesium chelation against DNTP concentration. We examined the thermodynamic battle against primer dimers, the photophysics of cyber-green intercalation for real-time quantitative monitoring, and the elegant biocomputational safeguard of the internal positive control.
We really went through it all.
Ultimately, we saw how manipulating these microscopic variables translates to macro-level clinical power, transforming how hospitals rapidly deploy syndromic panels to diagnose meningitis, acute respiratory distress, and global viral outbreaks.
It demonstrates how the rigorous, agonizing optimization of physical chemistry directly correlates to the preservation of human life and the integrity of epidemiological data.
The source material acknowledges the immense weight of this technical journey. It closes the presentation with a very simple quote on the final slide. The best way to learn